Iron is an activator of Wnt signaling. And Wnt and BMP are both dorsalizing factors. And actually the choroid plexus comes from the most sort of dorsal region of the telencephalon. And so simply by putting on this pulse of these dorsalizing factors, she was able to generate these organoids that look very much like the actual human fetal choroid plexus. And she could also demonstrate that these organoids show proper polarity and express things like the aquaporins, which are so important in CSF production. And that they also show typical features of the secretory epithelium. You see lots of microvilli, tight junctions, and these multivesicular bodies and extracellular vesicles. We also noticed that these organoids over time started forming these very large fluid-filled sacs. And just for reference here, the size of this dish is 6 centimeters across. So you can see how large these tissues can actually get to be. And when we leave these out for a bit and let the media turn really pink, you can see that the fluid inside is not just simply media, but some kind of clear fluid. So that started making us wonder if maybe these tissues were actually making a CSF-like fluid. So we extracted fluid from the inside and sent it for mass spec. And lo and behold, we found that the majority of the proteins that we detected were also detected in actual CSF samples taken from in vivo. So of the top 550 proteins found in the organoid fluid, 49 of them we also find in actual CSF. And a number of these are actually really interesting biomarkers, things like serpin F1. You can also see ApoE on here, for example. So suggesting this might also be an interesting system to look at biomarkers for different neurological diseases, including things like neurodegeneration. The fact also, let me just go back, the fact also that that the fluid inside is not simply media suggests that there might also be a specific and selective barrier forming here. So we wanted to look at that, and we decided to go for something really selective, but also a very small molecule to really test the system. So it's known that L-DOPA can cross the blood-brain barrier, but not dopamine. And the blood CSF barrier has to be just as selective as the blood-brain barrier because the CSF has free access to the brain. So we wanted to test if our barrier in these organoids was likewise so selective. And so we did this by NMR. We just basically added these to the media and then extracted the fluid and sent it for NMR and found that indeed L-DOPA was able to cross, but not dopamine, or at least dopamine to a much less degree. We also looked at some other drugs. So bupropionyl is known to cross into the blood, into the brain, and indeed we could see it entering, whereas methotrexate does not, and indeed we could see it wasn't entering. And if we place these different drugs that we tested on a curve where we compare with in vivo measurements, so this is the ratio of drug detected in CSF compared with plasma, and then this is our in vitro measure, we can see a very nice correlation. But not only that, the slope is actually equal to 1. So showing you that not only are these highly correlated, but it's actually quantitatively predictive of the permeability that you would see in vivo. And importantly, these measurements in vivo, these are taken from human. So we only really see this nice strong correlation and one-to-one relationship if we compare with actual human values. Of course, it's known that the blood-brain barrier is different across different species in terms of its selectivity. Right, so basically overall, we've been able to generate these choroid plexus organoids with nice selectivity and that produce a CSF-like fluid. Now, very recently, we also wanted to look at the SARS-CoV-2, the virus that causes COVID-19. And this is because we found that ACE2, the receptor for SARS-CoV-2, is quite highly expressed in choroid plexus. And indeed, we found that this virus could infect choroid plexus organoids. You can see this strong infection. And this led to a breakdown of the barrier where you can see Claudin 5, which normally very nicely labels the junctions between the cells. You can see it becoming more dispersed. And this also leads to a functional breakdown barrier where you can see proteins actually leaking across the barrier. And so we think this has important implications for some of the neurological symptoms being found in some of these patients. And it suggests that if the virus does enter the bloodstream, which of course is a big if and probably only happens in very severely infected patients, but nonetheless, if it does, these choroid plexus cells would be especially susceptible because there's really no barrier between them and the blood. These capillaries are fenestrated. And then these cells being quite susceptible, the barrier breaking down, that may lead to not only leakage of the virus across into the brain, but also leakage of neuroinflammatory and immune cells that could lead to neuroinflammation. So now let me move to the second part, looking at long-range connectivity. So this was work really spearheaded by Stefano Giandomenico, a previous PhD student in the lab. And he showed that if we take our organoids and culture them for longer term at the air-liquid interface, so we modify our culture paradigm here, where we actually slice the organoids into really thick sections, so 300 micron sections, and place them on filters with liquid below and air above, that we can actually get really improved survival of neurons in these tissues over much longer term. And so we end up with a really nice, dense neuropill reminiscent of what you would actually see in vivo, as opposed to whole organoids where we don't do this. And so this is kind of the reason you see this in whole organoids is, of course, because of the lack of vasculature. But by slicing these, we're able to get better nutrient and oxygen supplied to the internal regions. And this also allows for much longer term neuronal survival and maturation. You can see these nice pyramidal neurons here. And you can see nice formation of complex dendritic trees and dendritic spines. We also showed that these neurons are nicely functional. But interestingly, we also found that these cells seem to send out their axons in very specific patterns, so not just randomly. We find they form these large bundles, sort of reminiscent to axon tracts. And actually, some of these bundles seem to leave the organoid entirely and just travel over really great distances. You can see the scale bar here is 0.5 millimeter. So we can see some of these bundles traveling, over even up to 1 centimeter in some cases. And we can, look at them in different ways. We can look at their orientation, for example, but we can also even look at things like anisotropy in a sort of similar kind of fashion to what's being done for, MRI data for the human brain connectome. And so we can look at how these different tracts are connecting across these organoids and even away from the organoid. So we became interested in some of these externally projecting tracts because I told you these organoids are primarily telencephalic and mostly cortical. But of course, some of the neurons, a lot of the neurons in there are subcortical identity cells. And so in data that I'm not showing you, we showed that some of these externally projecting tracts actually do trace back to the subcortical identity cells. And one of the important subcortical identity tracts is the corticospinal tract, projecting from the cortex down into the spinal cord, innervating these motor neurons of the interneurons within the spinal cord, which then innervate motor neurons and then form neuromuscular junctions to activate muscle contractions. So we wanted to see if some of these externally projecting tracts we're seeing would be capable of innervating an exogenous target. And so we just provided that target. We performed explant cultures of mouse developing spinal cord with the muscle still attached, with a bit of the back muscle still attached. And when we do this, we see really nice innervation with these human tracts coming from the organoid and innervating the mouse spinal cord tissue. And then if we stimulate the human tissue here, so the human organoid is being stimulated, we can get muscle contractions in the muscle attached to the mouse tissue, suggesting that there is a functional connection there. You can see this is just traced here. And now if we cut those tracts completely, but leave the mouse tissue totally intact, you can see that these contractions go away. So this is being stimulated here, but you just don't see any contractions. And then if we look after the fact, we can actually fix and stain and nicely see these bundles projecting into the mouse spinal cord tissue and innervating it. And then finally, we've become recently very interested in axon, understanding the sort of ultrastructure of axons that allows them to form these really unique long-range connections. So human axons, especially these cortical spinal tract axons, are really very long and greatly increased in length compared with, for example, mice. And so existing data really in human axons is pretty limited to sort of classic electron microscopy, which gives you some insight, but it doesn't really have the kind of resolution. It's not really native, so you're not really able to see molecular resolution and organelles in their native conformation. But cryotomography So cryo-electron microscopy and then tomography allows you to actually see that. So we thought that maybe these organoids could be amenable to cryotomography, particularly these protruding axon tracts. And so in work done with Wanda Kukulski's lab, we were able to allow these organoid slices to grow some of these externally projecting tracts onto EM grids. Here we've placed next to them. You can see on the actual EM grid the growth of axons here on these grids. And so this is, we're able to actually then, by using correlative light and EM microscopy, to actually track individual axons and then image them by electron microscopy. And so here you're watching images taken every half a day. You can see the axons nicely growing onto the EM grid. You can see they really like the grid bars, but they do come off the grid bars every now and then and sort of move over to another grid bar somewhere. And then that's where we can then look at them. And so here you can see this nice big bundle, an individual axon now that we're looking at. And then we zoom in by cryo-EM and we can actually zoom all the way into a vesicle with its cargo here. So this is really powerful, enabling us to look at ultrastructure and even molecular resolution structure and trace it all the way back to the axon that it came from. And then this is just, so one of these tomographies, tomograms all the way through, where we've then also segmented different structures here. You can see a mitochondrion over here, various microtubule bundles, the ER, you'll see all of this being sort of traced out in just a moment. So here you can see the cell membrane of the two cells sitting right next to each other. And that's one of the beauty, that's one of the nice things about looking at this in a tract. You can really see the axon within its context. And then you see these microtubules in purple, the ER is in cyan here. And the beautiful structure of the ER wrapping around these microtubules in this really complex architecture. And so because of the resolution, we can really look at the molecular architecture and do things like looking at the orientation of microtubules within these microtubule bundles and show that they're all nicely parallel, just like they should be in an actual axon. And again, look at structure. We can see structures that haven't before been seen in the context of actual human axons. To look at, for example, these interluminal densities within the microtubule, as well as this very thin ER stretch here, where you don't even see any lumen within the ER, suggesting a primary role probably in this case of lipid transfer.